reason the population is expanding. Could be because of climate change. The individuals that will colonize the empty space will come most likely from the range edge. And it's this less dense area of the population. And so by chance, we're going to sample just some of these alleles. And so in this case, we're sampling a few more red alleles than the yellow-green ones and getting a higher proportion of red. And if this process repeats and repeats and repeats as populations spread, we can get the local fixation of this red allele. And this process is called allele surfing or gene surfing. And it's a process specific to species range expansions. And it's been studied quite a lot in Bern by Laurent and Stefan Peischel. And they've also looked at this mainly in the context of deleterious mutations. So this is just another image showing the same process of allele surfing, but with deleterious mutations. And this shows that at a range edge, you can get much lower fitness individuals arising on this local scale because of the surfing effect of mutations at a range edge. And so that's what this, the red and orange little pixels here are showing. These individuals are less adapted than these blue ones behind the expanding front. Okay, so that's why we thought range expansions could be of interest for this topic, because if you're an underdominant mutation, you're going to be negatively selected for or selected against when your initial frequency is small. But as soon as you get over a frequency of 1/2, you'll be positively selected for because you're pushed to either homozygote state. So in this case, if we have gene surfing at a range front, we have much more genetic drift. So you should be having underdominant mutations very much prone to this process, as the direction of selection could change and lead to local fixation at a range edge. Okay, so yeah, please interrupt if anything is unclear or if I go too fast. So to first look at this, we use a discrete space serial founder event model to understand what would happen just at one gene at the front of a species range. And so this is sort of the schematic here, where we have, just imagine this is the very edge of a species range at carrying capacity K. At some point in the future, it sends out colonists, so founders, and there's some founder number F to colonize the next front, the next empty gene. Then we forget about everything behind it and we just look again at the front. So this is now colonized with founders, and then we wait time t for it to grow back to carrying capacity. And then this repeats and this repeats and this repeats, and we're only ever following the front. We ignore any migration effects that could come from behind. That's an assumption of this model to make it a bit simpler. But the important parameters here are how long you have until you grow to your next carrying capacity before you then found again, and the founding size. Okay, so sort of jumping straight into results then. If we want to understand how likely an underdominant mutation is to fix at the range front, Stefan did some very nice math to explain this process using that discrete founder event model. And so we have here a figure showing the different fixation probabilities depending on the size of this founder event, so this F, and the time between events that you found, and also depending on the selection coefficient S. And so there's 4 different lines here for each different founding population size. And you can see that basically everything to the left of here is kind of a flat line lining up at the probability being 1/F. So anything that has a much smaller ST than maybe around 1 or 0.5, really what matters is only the number of founders you have. So this compound ST parameter is what determines your fixation probability. And when you're much smaller than 1, you basically are behaving like a neutral mutation when you're at the front of an expanding population. And so that means it can be quite likely, especially when founding sizes are small, that you can fix an underdominant mutation at the front of a species range. So now we know that it is possible and likely that these mutations can undergo allele surfing and fix at the edge of a species range. To look at, so underdominant mutations are likely to surf. And this is even more likely than deleterious mutations, which have, again, been studied quite a bit to look at expansion load in the past. I'll come back to the point I didn't say in a second in my head. So this is very easy for these mutations to surf at a range front. So then we wanted to look at this using forward simulations to see how often and sort of what the dynamics were. And so this is sort of my main part of this project. So we used SLIM, this forward time individual-based simulation program. and did a range expansion over a 250 dm landscape just in one dimension. And we have a mutation rate that allowed underdominant mutations to enter. And this was to look at the process of these mutations surfing and if it matches our expectation from this discrete model in the previous slide. I also will show you in a moment after some more simulations where we also force these mutations to exist at a range front to look at how they move after the range expansion is over, but I'll come back to that point. Okay, so for those of you who are interested, these are just the parameters in this first set of simulations. We have a quite small deme size, so 25 individuals per deme in each of these 250 demes across the whole landscape. And a decent migration rate, migration of 0.1. So this was sort of getting us somewhat close to a continuous space landscape because of this small population size. And then this is for underdominant mutations to enter the population. Every individual had a 10,000 base pair genome. We had some small recombination rate. And the important thing for the underdominants here is that heterozygotes had a fitness of 0.9 and homozygotes had a fitness of 1. So again, heterozygotes are not as fit as either of the types of homozygote. And so I'll just show you one replicate simulation as we go through time. So this is, again, the landscape, and then we look at the frequency of the alleles. It's just 2 because it's counting each haploid. So if this would be, when they reach the top here, they're at frequency of 1 in the whole population. And so there's several different underdominant mutations or underdominant alleles on this plot. There's a lot of them that are sort of segregating down here at low frequency. And here we can see there's one that's already surfed to very high frequency at the range edge. And so right now the species range has only reached here. So this is generation 100. And then I can continue through time. So 200, we can see the species is moving further across space, and we can see many more mutations increasing in frequency. So having these surfing events, we can see some of them also coming back down. And we can go through time. And eventually we get the population colonizing the whole simulation landscape. And I'll just pause there for a second. So there's many alleles that have surfed. There's also many that have not. But we can also now look at these Clines that have formed from the surfing event. So where we go from frequency 0 up to frequency 1. And we can see that if I go another point in time, they also are moving around in space. And I can go to generation 2000 and they've moved a bit more. And so there's a lot of different loci here that have undergone surfing events. And so I'm gonna, in the next figure, sort of take this same exact plot and sort of spread it out vertically so you can see the different mutations that have surfed to fixation. And each of them is now colored just alternating blue to orange so you can distinguish a bit. And I'm not sure how well you can see, but the black lines are generation 2000. And as you go to either lighter orange or lighter blue, you're going to earlier generations in the simulation. So if I were to look at this black line here, that mutation originally surfed somewhere over here, and then it moved through space. It's only sampled every 200 generations, I think, at each of these different blue lines, and eventually ends up right here. And you can notice that it's near a bunch of other mutations that are also kind of in that point in time. And there's some that are clustered here, and there's some that are clustered here. And so we noticed from these simulations that these alleles were not only surfing to fixation, but that the Clines would then move and then approach each other in space and sometimes align together. So we wanted to look more at this in terms of what was going on. A priori, it makes sense that fitness will be lower at the Clines and even lower when you have multiple locus underdominant haplotypes, but we didn't know what exactly was causing this process to happen or how. So we see many surfing events happen. We see these Clines drift in space and move after they've established, and that we see that they align in space as well. So why and how is sort of the next section of the talk. So I did another set of simulations. We have a slightly higher carrying capacity and a slightly higher migration rate. That's not super important. We just wanted to have things move faster so we could look at this movement without doing much more simulations. So it was like a computational limitation. But now there's no mutation rate. We just have at a range edge 5 loci that we manually say, okay, now there's an underdominant mutation, and we condition on it surfing so that we can get 5 Clines and follow what these 5 Clines will do. And same fitness scenario, heterozygotes at 0.9, homozygotes at 1. And this shows what happens to these 5 loci. So now we're looking at time on the x-axis, going through generation 0 to 20,000. And this is the landscape that we simulate. So it goes from 0 to 250, but I'm zoomed in just on where these loci are existing. And so each sort of line here is a different underdominant locus, and it's where the center of the cline is for where that allele goes from frequency of 0 up to 1. And so at the initial point in time, they're all at separate spaces in space. This orange one, it's just slightly different. So it has 2 lines. It's 1 locus, but it has surfed to frequency of 1, and it's also later surfed to frequency of 0. So it's like a bump in space. And so it has 2 clines for 1 locus at different points in space. And so then we go through time and we follow these 5 loci, and we see that they're all drifting around, kind of randomly up and down, but eventually these 2 get close to each other, and then they stay close to each other the whole rest of the time. And eventually these 2 get close to each other, and they also align in space. And then these 3 align in space. And if we did many more simulations, probably eventually they would all either drift off the edge of the landscape or together and be 1 multi-locus cline of underdominant mutations. I'll just show you a few more replicates to show you that's not like handpicked as a super rare event, but there are many, these are color different, but many different cases where we have these 5 loci and they're meeting randomly and then staying together. They meet and stay together. It happens here twice. So sometimes we're ending up after 20,000 generations with only 3 different clines, some of which are multi-locus. So here we have 3 loci in this one, 2 loci here, and still 1 that is alone there. But lots of different ways in which these clines move in space and align with each other. Okay, and so to get at this, to sum that up, so we do see surfing events, that they move after they establish, that these clines align in space, and it's only seeming to happen after they reach some critical distance apart. So they were drifting around for the majority of that time before, and even after they're aligned, they're really just bumping around in space. There's no pressure pushing them one way or the other until they seem to get close, and then there's something keeping them together and not letting them move apart. So why is that the case? And so here Antoine and Stefan also did some very nice math to sort of understand how we can explain the fact that these clines approach each other and align in space. So we'd have a model where we look at after a range expansion has completed and that these clines already exist independently of each other. For now, we're going to ignore genetic drift and just look at 2 loci in continuous space. Okay, so we have this 2 locus model of underdominance. So we have the A allele and the B allele, and the single heterozygotes are each underdominant. So each heterozygote is of poor fitness, these 4 panels here, and the double heterozygote is the most loaded, so the least fit out of all possible combinations of alleles here. Okay, and so that's what we look at through this model. And so Antoine initially just makes very simple L-shaped clines. So we have 2 clines here, 2 loci again. We have our wild type little a, and then some cline going instantly up to 1, and big A at this point in space, and then little b to the left of this red line and big B to the right. So these 2 different clines of A and B. And then if he moves the model forward, they sort of, this is a roughly drawn schematic. It's not actually mathematically accurate right now, but they eventually form like, a cline that's under migration selection balance where there's little b and big B coming from both sides, and it makes it, this curve instead of that straight line that was the initial condition. And then they let this go, and basically they always see that the clines will approach each other. There's going to be movement towards the center. There's one important, I didn't get to the caveat yet, no. So that's, if we think of what genotypes are existing, so over here we have little a and little b, so these are homozygous, these are the most fit. Same on the far right, big A, big B, these are also the most fit. And then in this area where we have the big A cline, we have heterozygotes at the A locus and homozygotes at the B locus, so some fitness less than 1. In the middle here, we still have homozygotes of both loci. So these are also a fitness 1. they're also still the most fit in the population. And then at the second cline, we have heterozygotes of the other type, little b, big B, but homozygotes, big A, big A. So 2 regions of low fitness and 3 regions of high fitness. But the important assumption that went into this model, which explains why these clines will move together, is that there was always some overlap in terms of the cline actually spreads all the way to the end, so that there is still some chance of this B locus migrating beyond. I hope it will become a bit clearer as I explain it in the next slides, but basically the clines were never at 0 or 1, they're always, because it's this continuous sort of infinite model, there's always some small amount of the other allele anywhere on the landscape. And I'll explain why that's important in a minute, because if you remember what I said in the simulations, those clines are always drifting around until they happen to run into each other. These ones, there is no drift. It's because we assume they can always overlap. And this is why they always move towards each other in this model. Do interrupt if it's not clear. But basically, we can look at, through time, what is happening at each of these clines and why they're moving together. So if we have no recombination, you remember we have the A cline first and then the B cline. So we're never going to get, we're never going to get big B with little a, but we can get big A with little b. And so if we look in space, no recombination going through time, we have this haplotype big A little b existing at some point as a single heterozygote, which is shown, so this is the frequency of that haplotype from where both clines exist, and this is where the single heterozygotes exist. You can see there's 2 peaks, and as we go further in time, they get closer to each other, and this sort of middle spot comes up, and we have an increase in single heterozygotes as the clines are coming closer together. So single heterozygotes exist at each cline. As the clines move towards each other, they form sort of one area. And eventually they're going to create double heterozygotes as they approach each other. And so then we see double heterozygotes taking over, and there's really no more single heterozygotes left. In this case, it's because there is no recombination that we don't have the single heterozygotes anymore. So as soon as these clines align, we have forced mating between the 2 homozygous types, and we make double heterozygotes that, even if they are very unfit and selection will be removing them, they'll still continuously be made from the mating of the 2 homozygous types. So even if the single heterozygote, little a, big A, BB is more fit than the double heterozygote, it's still gonna keep happening because of migration and mating. And so selection can remove single heterozygotes. And once they're gone, we're left with just the double heterozygotes that cannot be recombined away either. So they become the predominant form where the 2 clines are. And if we look at a few other measures of what's going on in the populations, we can look at load and linkage disequilibrium and use, as we would expect, the most load is where all of the heterozygous forms are, and then it's even greater when we have the double heterozygotes in the middle of the landscape. And as soon as we have these double heterozygotes forming without recombination, we have a bunch of linkage disequilibrium between the 2 loci. Okay, and so, I hope this will make sense. So I guess I didn't really fully explain why still the clines approach each other. If I did, it might not be clear. But if you think back to this diagram here, we still have both homozygous types on either end, even though we still have one in the middle. But basically, we have the little a allele is favored by selection on the far left, where it's in this double homozygous form. And it's going to always locally increase in frequency from the left. And this will move the big A cline to the right, because at the same time, the big B is favored from the right, and it's occasionally getting heterozygotes from the left. And so that's why it's going to move to the left, because selection will favor big B over big B little b. And so this is happening at both sides. Even if there was this center area with double homozygotes, there's the occasional heterozygote that makes it a bit further to the other cline. And that means it's on average less fit in the center between the 2 clines, so they move towards each other. This was maybe the hardest thing for us to explain when we were at this paper. So I think I say it again after we look at the case with recombination, if it's not clear still. So now this bottom row has free recombination, and it's similar but slightly different from the no recombination case. The major difference is that it's actually slower for the Clines to align with each other. And this is perhaps a bit counterintuitive. So you see nothing's really happening until we get up to this point in time. We still have a similar pattern here for the single heterozygotes. Once we have recombination, we can get this other haplotype, the little a big B, because it can be a result of recombination between individuals. We still have single heterozygotes because they can again be created through recombination while there's mixing across the Clines. We still have double heterozygotes just like we did before, and we still have load and LD. Do I explain it in the next slide? I don't remember. Yeah, so just thinking about with recombination, because we can create single heterozygotes now, selection is less strong against those because they're not as maladapted as the double heterozygotes. So that means the Clines will move slower because there's less pressure from selection as it's locally favoring the homozygotes relative to single heterozygotes instead of relative to double heterozygotes. So the selective advantage of being either little a or big B at either of the two Clines is lower. And so the movement toward the other Cline is slower. And it's also a related observation that these Clines are shallower where we have recombination happening because you can have single heterozygotes existing. Okay. And so then to sort of understand in more general terms what this pattern is, we looked at how fast these Clines should align to each other and what it depends on. And so here we have just if time is on the y-axis and how far apart they are is on the x-axis. This is measured in terms of diffusion and selection and also in terms of selection, but this is still more generations towards the top and greater distance apart towards the right. And for a bunch of different cases of recombination relative to selection, so greater recombination as you go towards the blue and stronger selection as you go towards the triangles, we see the same pattern everywhere. So the time it takes to align depends on the strength of selection S and on the recombination rate R and how far you initially are apart. That's the obvious one. The further you are apart, the longer it will take to align. But you can see that as selection gets stronger, from circle to triangle, it takes less time. Selection is stronger, forcing the Clines together at a faster rate. But as recombination is greater, it takes more time from the red to the orange to the yellow to the blue, because you're creating more single heterozygotes as the Clines are moving along, which is basically reducing the efficacy of selection against individuals in your population. And the further you are apart, of course, the longer it takes you also to align. Yes, okay. And so to look at this, perhaps this is another way to look at it a bit more clearly. If we have no recombination on the left and free recombination on the right, and we just, again, look at two different Clines, the A and the B, these are at different time points from the dashed lines to the solid lines over space, and this is allele frequency. And with no recombination from, 0 to 400 generations, we can see quite a lot of movement of these Clines towards each other. So they're starting out here at the smaller dashed line and then moving towards each other and ending at the solid line. In the same amount of time, with free recombination, you can barely even observe that there's any movement at all. The lines are still all overlapping with each other. So it's quite a difference that these are, of course, the two extremes of recombination, but it's quite a difference that recombination makes. And so over 400 generations, these Clines are coupling without recombination, but they've barely moved at all when there's free recombination. Okay. And so probably you're thinking, well, what about your assumption of these Clines always overlapping? This is not realistic at all. I also said this at one point to Stefan and Antoine. They're like, yeah. But so like I said, in the simulations, these Clines are just drifting around until they happen to get close enough. So there is some critical distance apart at which they will eventually overlap and then align. But until that point, they are just drifting in space. They're at migration selection balance. If fitness is one on either side of the two Cline, fitness is one on either side of the Cline, there's no reason for selection to move it one way or the other. So if we were to relax this assumption in the deterministic model and allow there to be drift, we can sort of evaluate what this critical distance is. It's specific to the carrying capacity also, so we just applied it in this case to the simulations I showed you, so carrying capacity of 50. And you come up with an equation that includes migration and selection, saying once you're this close together, then you should be in this realm of the clines starting to attract each other, because it's basically depending on how shallow or steep your cline is at one point. I can also do this towards the video. Like if this cline reaches 1 here, but it overlaps with one that's not yet at 0, then they'll start to align. But if you're this far apart, there's no interaction and there's still this region of high fitness between the two clines. This comes out to be 10 deams in our simulations. But if we actually looked at the critical distance in the simulations, we actually see them get as close as 4 deams before they always will couple. So I'll explain that a bit more now. This probably looks super confusing, but this is again time on the x-axis. And this is the distance between the centers of any 2 clines that are next to each other in a simulation. It's a bunch of simulations thrown together. And basically what I've done is, if you can see the light blue being different from the dark blue, light blue distances are clines that never couple. And if they're dark blue, that means eventually they're going to couple, which means they're going to go down to 0. The distance between them will become 0. And you can see there's a bunch of dark blue ones up here all drifting around too, and eventually they'll still all go down to 0 here. There's a bit of noise around 0 at 1 or -1, because defining the center in the simulations was a little tricky. These were very steep clines, and so to say where the center was, I took the frequency closest to 1/2, but sometimes it was like 0.6 on one side or 0.4 on the other, and so the center is basically plus or minus 1 deem for the center, which is why you can see some being -1 distance apart. And these reddish, yellow, orange lines here are just saying how far apart to sort of better visualize it. It starts at 3 deems apart, 4, 5, and 6 for the 4 different colors. And all of the dark blue ones, none of the light blue ones go below 4, which is how I can say they can get as close as 4 deems without coupling. And that's just an observation from this set of simulations. Maybe we would get a closer critical distance if I did many more generations or many more simulations. But the point here is that in practice, this critical distance was much smaller than what was predicted from the model, even when we allowed for drift. So there are important effects of drift that are not accounted for in the deterministic model. Okay, I think maybe there's one more point. So this next slide is sort of also a future direction, but we investigated a bit in this paper. What happens if you still have underdominance, but one of the homozygotes is more or less fit than the other homozygote? And so in this case, there should be selection towards one of the homozygotes and still away from the heterozygote. And here we see, how do I best explain it? So ignoring anything else, if we have one homozygote more fit than the other homozygote, selection should go in favor of that one. That should be quite apparent. So the cline should move over space. And here we're looking at allele frequency on the y-axis and space on the x-axis. And if we look at, which one first? Let's look at free recombination first. This is our cline that's going to move. So there's some favorable homozygote on this side of the cline. So this black line moves over time. This dashed line is where it started. It's moved to this second cline. This second cline is just neutral in this model. And it's actually going to go past that cline, but it pulls that cline a little bit off of its original location in that dashed line. So the red cline has moved a little bit because the black one ran into it, met it, maybe aligned for a little bit, and sort of had this some linkage disequilibrium keeping the two clines together. But because there's free recombination, it eventually escapes, and this black one will keep going. If we have weak recombination, I think recombination might actually be 0 here. Again, we have our black cline moving towards this red one. It meets it, and it fully aligns with it, and basically it's grabbing that cline and moving it through space as a multi-locus or as a 2-locus cline, where only one of them is being selected against. So that's depending on the recombination rate, whether the clines can sort of escape each other or not. And this is something we want to look at a bit more, because maybe it is more common that there's not this perfect balance between homozygous types, even if there's underdominance for heterozygotes. But so it seems like there can be a lot of different dynamics in terms of what recombination is going on in how these clines will move over space and how they will interact with each other. Okay, yes. So I think then, yeah, I have some conclusions. So I think this sums up everything that we saw. So underdominant mutations can surf on the front of a range expansion, and they're more likely to establish than deleterious alleles. They can be maintained in Clines, and these Clines will drift over space up until they reach some critical distance, and then they can attract each other and align in space. This can happen and create double or even triple or greater heterozygotes at these aligned Clines. And one of the sort of bigger points we allude to in this paper is that maybe if you're getting many locus underdominant Clines aligning, you'll have sort of two different haplotypes or genetic types on either side of this Cline. And this could be maybe some early form of speciation or some sort of hybrid zone where you're really selecting against any crossing into this area of low fitness in the Cline. And it could happen without any sort of ecological selection. It could just be the result of a species range expansion over space as underdominant mutations surf. Yeah, so the Clines do align when there's this overlap of unfit single heterozygotes, such that double heterozygotes are produced. How fast it happens depends on recombination and strength of selection. One point I forgot to mention on this slide is that, so personally having worked much more on like deleterious mutations and their surfing in the past, that has always been a transient phenomenon, that this result of a range expansion leading to local fixation at a range front can be deleterious, but it can be erased as migration catches up. The interesting thing here with the underdominant mutations is it's not something that will be erased after the range expansion is finished. As we've seen, these Clines will then just persist in space because the individuals on either side are still doing fine fitness-wise, and they can then lead to these interesting dynamics that could persist over long periods of time, perhaps throughout the existence of whatever species it is. And then I think, yeah, I just mentioned a few more future directions. So one interesting thing we also haven't looked at is the impact of epistasis. So what if there are interactions across these different loci? This could create some really interesting dynamics in terms of how and when the Clines form, and if there's sometimes what negative epistasis or positive epistasis, or what will happen when we get these multi-locus Clines aligning. And then we want to look a bit more at this unbalanced fitness between homozygotes and whether this capturing of other Clines and moving with them can result in some other interesting things. And then maybe one of the fun things to talk to some people here about today was how we could ever investigate this empirically. One other thing I should mention as I guess a major caveat is we don't know how often underdominant mutations arise. So if they arise a lot, this could be super important. If they're quite rare, maybe it's not so important. But one good example that would probably be most interesting is that inversions where you have the heterozygote form being maladaptive could be a major way where we see underdominance at, it's not one locus, but at like a region of the genome. So looking at this in inversions could be maybe the next best empirical investigation into how important this process might be during species range expansions. And so, yeah, otherwise I think identifying individual loci that we know to be underdominant is probably a very difficult task to find on a broader scale across many species. I think this is all. And so with that, yeah, I think it's better I stop. So I have to thank again Stefan and Antoine, who did a lot of this work as well. And then we had some feedback from Nick Barton and Nika Polikova, as well as Laurent Scopier, and then Bern and the SNF some funding. And then I'd be happy to take any questions if there are.
So it's nice to hear some real claps.
Yeah, that's also a different thing I've not heard in a while.
Virtual ones. So, all right, so we already have a few questions online, but are there questions in the room first? Mathieu has a question. Okay. Try to say it loud and
yeah, I can also maybe speak better into the microphone.
Perhaps you can put the mic, your sound loud too, so that people online.
I think with this I don't have.
Okay.
Or maybe yeah.
Oh, okay. So what's your question?
It seems to me that thanks for your talk. It seems to me that the situation you're modeling is where all four homozygotes are equally fit. And I was wondering, in a real ecological landscape, what kind of situation would that be?
When there's two underdominant, like perfectly underdominant loci.
Yes, because I think, you know, on an ecological gradient on.
Oh, I see. Yeah, there's no underlying like landscape difference here. I think this is what you're getting at. So like if there was a changing climate or something like this and it changed fitness, it's not accounted for here. How it would change things. Yeah, you would, even in that scenario, you would have probably moving Clines for whatever type is, well, it's worth investigating probably because you would be favored in maybe only a very small region of the landscape and you would stay in that region. And so maybe this would cause all the Clines to come together at one point in space, depending on how the landscape changes. Yeah, there's nothing else going on that would cause, there's no selection that changes over space at all. Yeah, exactly. But yeah, it's something that we should also, I should add that to the future directions because it would change the dynamics, I think. And if there were any sort of certain like bumps or anything that's not a continuous gradient would probably cause the Clines to maybe align in a certain spot rather than just randomly due to drift.
All right, so there was a question by Thomas, so we're going to try this. So it means I have to put the sound on my computer. Thomas, tu nous entends? Can everyone turn off their microphones, please? Sorry, this.
So do you hear me? Oh, yeah. So so I was wondering. So you had some feedback from Nick Barton apparently on your work, and I'm sure he mentioned that underdominant Clines are known to attract each other. That's a theory of been shown several times in theory for tension zones. And this occurs respectively of range expansion. It occurs in a continuous space. And so have you looked at this theory? Because it's really similar what you find from these previous results. I don't hear anything back, but.
Okay, so you didn't hear me. I should start over the answer.
Yeah, I didn't answer.
Yes. Yeah, so it's true he's done this like without the concept of range expansion. And so we were interested in if that changed anything. And I don't remember the exact details of how different it is from what he found. But yeah, he pointed out to us, I think it's mostly in like also in one of the appendices of his paper that he has something quite similar to us. But still different enough that we wanted to pursue this. I'm sorry, was there more to the question?
No, because it was occurring, I think it occurs irrespectively of range expansion.
Yeah,
it seems to be completely irrelevant.
I think it's if the Clines already exist. And I think one thing we wanted to add was, can a range expansion create the Clines to exist at all?
Right. Yeah, okay.
That we add, yeah.
Thanks.
And yeah, so it's just one more way to get the Clines to even exist in that space.
Okay, so yeah, I had somewhat of the similar question as Thomas. So basically, the phase where the Clines converge is not during the expansion.
Yes.
So what the expansion does is provoking a series of Clines that are more or less close together, and then this will trigger this mechanism where they align, right?
Yes.
Okay. Another question in the room? Yes, many questions in the room. So students first. So the question, sorry, I repeat, if the Clines can eventually uncouple after they have coupled. The stability, something like this?
Yeah, so I think we didn't investigate the whole, I should turn my microphone back on.
Yeah.
Okay, but I think it makes an echo. Okay. We didn't investigate all the possible parameter space, but like I showed in this last one, there was one neutral Cline and one under selection where it does either couple or it moves past and uncouples depending on our combination. And so even if that second Cline wasn't neutral, I think if we had a low enough selection parameter, it could also uncouple. So I think when we have really weak selection and really high recombination, it's a scenario where they could move apart. I don't know exactly where the border is in terms of the numbers, but I think that is what we would expect to happen at least.
So who else had a question?
Yes, let's put my microphone. Yeah.
Yeah, so we didn't yet think of this.
So
is the movement because there's more migration proportionally? Is this the, okay. Yeah. Yeah, I mean, I could certainly see that this would lead to them to move. And then I guess depends on which side is the more dense one in terms of when they would either meet and stop or like meet and keep going even more strongly than before. Because I guess you could reinforce higher density on one side versus much lower density on the other if they both have like that homozygote being better, or it could be a stopping force. But even if you have on either side like the density difference and they're going towards each other when they meet, I don't know. In some cases, I guess they could even uncouple and go past, or they could couple and.
If you have a trough and a population density, they are really blocked. It's another pattern.
Ah, it is. Okay. Okay.
So that occur in the simulations? Did you see some trough where there was a
no, so the simulations, the growth rate was high enough that we don't have like a large trough where the Clines are. I think, well, I mean, I would assume at some point if we had like 5 or 6 loci aligning, then eventually the fitness would be so low that we would observe this trough regardless of our growth rate. But yeah, it's not something we investigated to that depth yet either. But this would, I mean, depending on the width of the Cline is whether it would block or not. Right?
I was talking about exogenous factors that control the density.
Ah, okay. So like if the landscape can only support so many.
Yeah, and I think this relates to Oliver's question, doesn't it? Oli?
That was your question.
Oh.
You, Oliver?
Hello, can you hear me?
Yes, I can.
If someone.
Yeah, so my question was related to the issue of the ecological gradient. And I was more thinking about more from a demographic perspective, that often range expansions are viewed in sort of in a scenario where you have a core, a big core and expanding front with small populations. And I was wondering how a more constant flooding from the core could influence the drift, which is driving the evolution of the Cline, no?
You're muted there? Or?
No, this one is on.
Okay, then I talk into this one. Yeah, it's a good question. And I think, I guess there's a couple answers. So I mean, for what we showed here, you need the allele surfing to happen. And if from this first figure, if I go, this first figure that showed you the likelihood of the underdominant mutation fixing based on the number of founders, requires a small number of founders to be, like the highest we get is what, 20.20 for even the smallest number of founders. So as you increase the number of colonists, so like what you're saying with a higher migration rate, you have less likelihood of forming the cline in the first place. And so, yeah, it's entirely dependent how likely they are to form in terms of how much surfing is happening, which is based on how much drift is happening, which is based on how many migrants there are moving to the front. If your question is more like behind the front, migrants come and catch up, by then the clines would have already formed if there were still small colonists at the front. And then the dynamics should be what we showed, like also what Nick has shown in the past in terms of the clines moving and or coupling. But I guess it depends on the shape of your expansion front, how steep it is in terms of where the small number of individuals are. And if there's in sort of the topic of range expansions, there's people who study these pushed fronts versus pulled fronts, and it's whether your migrants come from behind the front to colonize further, or if they're really the ones right at the front moving ahead and eventually migrants catch up. And I can imagine in this pushed front where the migrants come from further back, there's probably less drift, and maybe it's less likely also to have this formation of the cline. And so again, here, this was just only looking at the front beam as the colonizing beam. But in the simulations, it was also a stepping stone. So you could only go one beam at a time. So with long distance dispersal, I think in general, surfing is probably less likely like this, and this would hold for expansion load sort of things too, not just underdominance. Like whether you get surfing or not depends on your migration and shape of your expanding front. I hope that answers.
Yeah, thank you.
Okay, so he was here. More questions in the room? Okay, so if not, I think we're going to end the seminar here. So let's thank Kim again. And so we'll see you next week with online only.